Hello, everybody. I hope this works now. So, welcome. I'll tell you something today about how to make sense of lipidomic data. And yeah, first, we wanted to tell you quickly about Lipotype. Uh, we are a company providing lipidomics services, but also strive to use lipidomics for a better life. And we have actually many lipid services by now, but like the core process that we initially used was shotgun lipidomics, which we did automated sample extraction, automated sample infusion, and then MS analysis. And later, there is data analysis of the mass spectrometer data by the Lipotype Explorer, which then turns that into values. And then you have, for example, Lipotype Zoom or other products turning that into insights. And that's actually what you want, because what I thought is actually the best thing that can happen is that you get your lipidome, that then some matching happens and out comes a small number of lipids that predict your outcome, and is clearly linked to something actionable like a druggable protein. That's, of course, the dream, but let's see how far we can get. So what we will do today is we look at how data might look like, how we can clean the data, and how we get to a first result. This is more a seminar saying, okay, how can we do a first analysis and preparing later analyses, and then looking first at the first basic questions that arise. So what you get is usually a matrix of data. So you have a feature, which is kind of a lipid name, and you have a lot of samples, and then measurement of these samples. And of course, doing lipidomics, these names mean something, and these are codes for lipids and lipid species. And there are multiple categories we'll get into later, but the most common one is a glycerophospholipid, which is the signature is basically a glycerol backbone and a phosphate, to which a head group is attached. This is the most commonly known, that you have a phosphatidylcholine, where you have a phosphocholine head group here, or etanolamine, or phosphoenol is inositol, for example. This is only part of the variation that we're having. A large part is also due to the fatty acid chains that have a range of carbon atoms, so they can be 12 carbon atoms long, or even 22 or longer. And they might have different numbers of double bonds and position of these double bonds. You might have heard of omega-3 and omega-6 type fatty acids. This is then the position of the double bond. And if you take this all together, you could actually end up with about 9,000 individual lipid species that can be produced. This is usually not what you're getting in your full lipidome, because nature and the specific samples you're working with are usually restricting that into specific lipidomes that mean something in the outcome. If you look at the specific plasma lipidome, this is what this might look like. You have the categories in the middle. What we just talked about are the glycerophospholipids. These then have different lipid classes, and they can then be a species or a subspecies. And I would actually like to take a moment to introduce this layer of species and subspecies. So basically, how much do you know about a molecule when you're doing lipidomics? This is from the Swiss Lipid Database, which is a service we actually quite like, because for lipidomics, they exactly make this hierarchy of information. So what we just talked about, there's the category, let's say the glycerophospholipids, there's the class, the PC or ether-linked PCs, and then there might be a species. And for the species, you know the total number of carbon atoms and total number of double bonds in that molecule. That's kind of this layer. If you also fragment the molecule and actually get the fatty acid information, you might know the total number of carbons and double bonds within the fatty acid. Then you actually go down to the molecular species. So this is a 24, which might be a arachidonic acid. If you also know the position of the fatty acids within the glycerol, so there are two, so this could be one or the other way around, then you're actually talking already about the structural subspecies. And if you, in addition, know every position of every double bond and its conformation, so if it's an E or Z conformation, cis or trans, then this is the isomeric subspecies level. I'm pointing this out because usually in databases, when you look at molecules, you either find this level, or if you're going, for example, for KEX and pathways, you usually find the class level. However, what you measure with lipidomics is usually something in between species, subspecies, structural subspecies. Getting a jump to isomeric subspecies is then a technical, more sophisticated jump. Good. So next is we go for a little bit of data cleaning, data wrangling. And the first thing you actually notice is that the data that you're getting is having some blank spots. So these are usually NAs, which means true, in this case, true NAs, because we do not know what the value is there. So there's multiple ways you can deal with that. Sometimes you need to deal with it because some later analyses need a complete data set. But one point is you can impute them. And there's multiple algorithms. We actually recently did a little paper on testing most of these algorithms that came to our mind, and we actually found one and two which actually work very well for lipidomics analyses. And the other thing you can actually do, and most people in the field do, you actually work only with the highest abundant lipids in the data set. So that if you have the occupation down here, which is these lipids have been measured in every sample and these have been measured in only a few samples, then what people usually do, they say we accept about 70 to 80% of, well, the other way, we actually measured 30 to 20% of NAs. This is what we accept. And it has to be in about 70 to 80% of the values have to be there. And then you continue on with working with these lipids, even if they might be missing in one or the other sample. So then another thing that is always good to look at is your total lipid amount, which is simply the sum of all the lipids in your sample. And this is important. For example, if you look at this distribution, this is the total lipid amount on the x-axis and how much you're having that on the y-axis. You notice that there is a nice distribution of these values around here, but you might actually have a few that have not been correctly measured, and that's something that should be discarded. Further on, if you look at this total lipid amount and it looks like this, that, for example, sample 2 in this case has low values and sample 1 is higher and so on, then your later tests might be confounded. Because if you imagine you have sample 1 and you have an additional exactly the same sample 2 that doesn't have any changes but it has double the amount measured, then everything will have a factor of 2 difference, and you have a lot of significant hits, although the sample was not different. So it's actually quite important that you standardize, and best you standardize before you actually even measure. And that can be done by either measuring the protein amount you're having, or you can count the cells in the fax machine and say, this is the number of cells I'm providing. Or if that doesn't work, we often do that as an afterthought by doing the total, we standardize to the total lipid amount. So you actually end up having something which is called the molar fraction. And this is usually quite helpful in the sense that you standardize it. It has also its downfalls, but that's usually if you have extreme changes within a sample, then this is also something that can happen. Next, what we like to do at Lipotype is that if we have a lipidome, the first thing we do is a principal component analysis, which is an unsupervised dimensional reduction. So what you're doing is you're taking that full lipidome with hundreds of dimensions, say mean hundreds of lipids, and use this dimension reduction to project it into a, let's say two-dimensional, in this case, two-dimensional space. That helps you to just get an overview of what's happening here. And the first thing you look at usually is the scatter plot, the score plot, which is a scatter plot of your samples. And here you would actually see that samples are nice, like replicates of the same sample are actually nicely clustered together, and they're separated by some greater distance. So this obviously then tells you, okay, this experiment looks like it would have been gone well, and this is a workable, I can continue working with this data. We've here had an example where we actually measured a screen and had many samples, which we measured at different cell densities and standardized to total lipid amount. So the lipid amount was in both cases 100%, but we noticed that if we took both of these groups from the screen into the same PCA, that the major difference, which is the first principal component here, was actually showing a large separation between the denser cells versus the not so dense cells. And that clearly said, okay, this is a major confounder that you're having in your data. And we ended up using only one density for the following experiments and not mixing them. But there's also other points where you usually easily notice by just glancing at a score plot, saying, okay, this shouldn't be the case, or that's a good sign. The next you can actually look at is your loadings plot, and your loadings plot gives you information about which lipids are actually responsible for making this separation. So every value on the x-axis here is basically the contribution of that lipid to that x-axis here, which is separating your cohorts. And what you're looking for here is actually patterns in the whole space. And what you notice if you have a keen eye is that because we have a enzyme that is responsible for ether lipid synthesis knocked out here, then you see that the circles, for example, the green circles are all on that side, while all the green squares are on that side. Doing little distribution curves over there usually also helps spotting this. But yeah, you're actually looking for patterns that jump out at you in order to distinguish, to basically see if there's something general in the data. What you need to do a PCA is actually imputing the missing values. We usually advise against replacing these missing values with zeros, because that will simply enlarge your differences. What usually is quite nice is imputing with the median, because it pushes things rather towards the zero in both directions. So it's making a conservative contribution, or using one of the algorithms that I mentioned before. Also, very important is to center and scale your data. Centering means to remove the mean from every value within this feature in this case. And scaling means that you standardize the variation, the standard deviation to 1. Why is this necessary? This is because the contribution of the lipid to the PCA is dependent on the abundance you're having. And if you, for example, have a plasma lipidome, you have a lot of lipid species with different abundance, but for example, cholesterol can be about 30% of lipids, so major contribution. And then everything that you do in the PCA is usually skewed by the value of cholesterol. And you rather want to have a fine-grained contribution of all of your lipids. So sometimes you see something nice like this, say, okay, this is what it is. Sometimes your PCA might also just look like this. So we have a lot of overlaps and you couldn't tell anything from that. So the major variation, this is what PCA measures between the samples, is not causing a separation, but you might not be interested in the major variation. You might be interested in some variation that is different and what you're looking for. So some minor lipids might also change, or some not so minor, just not the most abundant ones. And that is probably what you're looking for. So what I told you here in the first part was actually how to handle missing values, check total lipid amount, and get an overview of your lipidome by using some dimensional reduction like PCA. So next we're going to compare samples. And what you usually need is two groups, like here, the donut and the broccoli, or usually more the control or treatment. Or example, I will use for plasma is male versus female. And you need to choose a statistical test. And for this, you need to know that lipids are often not normally distributed. And what you can do here, I think data comes later, I guess, but basically you can also transform the data, basically, for example, making a log transformation, which usually helps, but often this also doesn't rescue all of the lipids. So what we usually end up doing is using non-parametric tests when we have enough samples. If you have only a small number of tests, you actually have to use the t-test because that's the only one that works down to 3 or even 2 samples. But then you often have to rely on the robustness of the t-test because you're not actually matching everything that the t-test requires. What is also always advisable is actually using paired designs. They have higher power because they're pairing, for example, paired means that you, for example, measure the same person before and after, and then you can assign the statistical test such that you actually look within the person. Have all people increased versus have both groups at a significant difference. And this is actually very important, especially in lipidomes, also in plasma lipidomes, because there's a high interpersonal variability in humans. That means humans per se are already very different in their lipidomes. And so it's rather difficult to find small changes for your two cohorts if they're already that different. And paired designs remove that variance between people, and then this helps you with your power. So, for example, usually you have hundreds of subjects in cohort studies where you compare two cohorts, and then you might have 30 or something if you have crossover designs where you can do a pair test. And also another point, dear Jimmy, is kind of if you just look at cell culture, cells one day versus the other day versus the next day, you see drifts that you can't control for. So always when you do experiments, see that you treat your cells as similar as possible. For example, if you're doing a screen, not doing the control at the beginning and then doing all the screen samples for the next month, but have a control with every treatment that you do. And this helps a lot by reducing the drift that you see in cell culture, for example, over time. So what you're then getting is for example, a volcano plot. This is usually it's a list of your lipids with some p-values and fold changes attached, which you can actually nicely plot as a volcano plot. This means the change, the ratio of change, the ratio of means is here at the x-axis, and the p-value is then on the y-axis. And this is usually plotted as the negative decadic logarithm of the p-value, simply meaning that if something is 10 to the power of minus 30, it might end up back here rather than down there. So the more significant is up, and the more important the change is to the right or to the left. So this is all your lipids here in that plot. What you're interested in is then, are there significant differences? So you apply the p-value and say, usually you have a cutoff of 5%. This blue circled ones might be all the ones, are all the ones that are significant. But one important thing when you're dealing with omics is that you actually have to control for multiple testing. This 0.055% rate is actually, for type 1 error, actually means that if you do 20 tests and there is no effect there, then you might nevertheless find one just by chance. And if you're doing hundreds and hundreds of tests, then you're bound to find some of these to be positive, so below your significant value, although there is no difference at all. And this is what there is a number of algorithms that can then correct for that. And what is usually used in lipidomics is called the Benjamini-Hochberg, the false discovery rate correction. So that moves the significant value slightly higher. We'll see later a better example. So the 0.05 would be at 1.3 here, and then lifts that up. So these are the ones you usually can trust. And then next is, okay, that's a list of lipids. What can I do next? What always helps is to now infuse a little bit of your lipid or domain knowledge. And if I go and say, okay, here I have all lipids that are higher in males, and here I have all lipids that are higher in females. If I just color them by lipid class, I find out that males have higher triacyl and deacylglyceride values in this cohort of 1,000 people. But I also would find that the females usually have higher amounts of sphingomyelins. Not particularly different, but statistically very different. And if I even look closer and have a look at which species there are different, you might notice that there is always a 2 at the second position, which means there's 2 double bonds, which are lipid species known to have a specific long chain base, which is called sphingadiene, which is higher in a lot of samples that are of significance, and also in females. Jumping gears, looking at another volcano plot. This is a knockout sample that we have a control here and have a sphingomyelin synthase knockout on the right-hand side. Sphingomyelin synthase creates sphingomyelin from ceramide using a PC and chucking out a DAG. And yeah, you might, with a keen eye, see that there is sphingomyelins here on the control sites which are higher, which makes a lot of sense. But sometimes this, if you're not exactly knowing what you're looking for, it might be a bit challenging to find it within the sea of individual changes that you're having. So one thing that we actually like quite a bit is looking at what we call aggregations. That means that you sum up specific groups of interest into new features. So you're doing feature creation, and that means usually you define what you want to sum up, within which group you want to sum it up, and what you want to standardize it to. So basically, for a class profile, for example, we would sum up all samples, all lipids within a sample that have the same class. And if you don't standardize and just take the picomole values, this is what we call the class profile. If we, in addition, also standardize it by the total amount of lipids, then we get the profile by itself, the class per sample. And if you look at this one now for what I just told you, the sphingomyelin synthase knockout, then this is what it would look like. You would actually get, for the free samples that we have, a bar or a value of the sphingomyelin synthase knockout and one for the control. So basically, you're taking all the lipids within one class and put the value into the class. And if you look now at the biology, what is happening, so the synthase of sphingomyelin is reduced, and we nicely see that sphingomyelin values in these cells were dramatically reduced. But we would also expect that if this arrow doesn't work anymore, that ceramides should be increased, and we actually also see them moving up a little, not that much, because what actually happens is that ceramides are strictly controlled, and that all these lipids that, or we suspect that all these lipids that would have moved down to the sphingomyelin actually now move down to the hexacyl ceramides, which the glucosyl and galactosyl ceramides, and also that you see. So the hexacyl ceramides are increased in this case. So this makes quite some sense. And all the other changes you might have might be actually ripple effects. So if you mess with sphingomyelin, you actually probably get something in the ether lipids. But yeah, these are often things that are more complicated and more sophisticated because the whole lipidome has to react. Just another example is, for example, the total length per class. So we take for these lipids how many carbon atoms we are having, which is the total length of the fatty acids. And if you look at another enzyme, the ceramide synthase 2, which is responsible for creating long chain ceramides, you actually see that in these sphingolipids, these are strongly decreased, while the short chain ceramides with about 16 carbon atoms are increased. Yeah, so these are basically some of the aggregations that we apply. Others are then also based on fatty acids, as I show you later. Good. So if you do univariate analysis, so basically looking at every lipid individually, you need to choose your test. You should examine your results, for example, with the volcano plot. Of course, account for multiple testing, and then you can explore more general features of your lipidome, let's say aggregations or sums. Another analysis we like to do is correlation. And what that means is you come up with another numeric value you are interested in. That could, for people, be, for example, the BMI or the age of the individual, or you actually treat it and you have a drug concentration that you're looking at. And that concentration might be, for example, here on the x-axis, and then this lipid might be on the y-axis. And what you're looking for, is there a significant trend that you're finding? So the more drug you use, the more this lipid is increased or decreased. And methods that you obviously use are correlation analysis methods, but there might be linear versions of that where you see if things move at a straight line, which is called a Pearson correlation. There might be monotonic ones, which is, for example, a Spearman's rank correlation. So things that have to move on a line, but have to move up continuously. Or you're actually dealing with multiple covariables. So you actually would like to see the BMI, but you want to correct for age. Then you actually need linear models, which are not as general as the Pearson correlation, but this also works. So I cheated here a little bit. I took the correlation of cholesterol with other lipids, which is usually giving you nice results because lipidomes in general are usually highly correlated. So for example, triacylglycerides with themselves are usually showing a lot of co-correlation. Here we actually took 100 plasmolipidomes and looked at what cholesterol, how they correlate with cholesterol. And sanity check, of course, cholesterol does correlate with itself perfectly. That's what you see here. But there's also a lot of other lipid species that show strong correlation with cholesterol. And here you have a histogram of first the Pearson's correlation coefficient on the x-axis, and then how many lipids correlate with. Here, for example, you see which ones there are in this forest plot. So as you see, this is mostly sphingomyelins, some saturated PC and cholesterol esters also. And of course, the same applies here. Now you would be interested, is there something general? I see a few sphingomyelins here. Is that true for the sphingomyelin class in general? And then you can actually look at the correlation of the classes as well. So there might be sphingomyelins, PCs, cholesterol esters, ceramides. These are all highly correlated with cholesterol. Or you would be looking at the top fatty acids within these complex lipids, mostly saturated, these like 3 up here, saturated fatty acids, 18, 17, 15, yeah. Good. So for correlation analysis, you choose your correlation type that you're interested in and examine also the correlation coefficients, the p-values, but also scatter plots, forest plots, and then more general sums that you can do. So, and finally, if you have done that and haven't found something specifically, there's also another method you can apply after you have done your analysis, which is called enrichment analysis. So basically, you have done a univariate result, or you have done a correlation analysis, and have now a set of lipid species that are different, but you can't find the pattern yet, and that's what enrichment analysis is for. And we always portray this with the basketball players. You have the nice Lipotype jersey, but yeah, you might ask why basketball? The thing that we always use this example to actually talk about enrichment analysis. If you imagine a school that has about 100 students, and you just say a third of them are tall students, and there's a third of middling students, and there's a third of small students, and you all tag them with different labels. And then you would look at a subgroup. In this case, it might be the subgroup of the basketball team, and do enrichment analysis. You might find, based on these tags, that the basketball team is usually enriched in tall students. So the smallest ones have a hard time competing. So usually that is the case. And this is simply how enrichment analysis works. So here we don't do tags like tall, middle, and small. What we do here is, for example, in a feature enrichment, we would give tags like this is this lipid class, or it has this fatty acid, or it belongs in the pathway enrichment analysis to that pathway. Or we also have reaction enrichments where you get more fine-grained information. Then this lipid would be part of that reaction, this and that reaction, so to speak. And then there's all kinds of tags that you can have. And then you can do the enrichment analysis. And how this works is simply you choose a background, which would be all students, 100, and tall students I have a third of. And this is the background, and the foreground, you would say, I have actually 8 players, and 7 of them are tall. And then you can use this overrepresentation analysis based on a Fisher exact test, which you need these 4 numbers for, and that would tell you if there is a significant enrichment or not. So if you now go for enrichment in a lipidomics context, that result I've shown you before, you have all these individual lipid species significant. You now need to choose a foreground, something you're interested in. And we actually like not only to, just go back real quick, not only choose everything that is significant versus everything else, no, everything. We actually like to choose what is significant usually in one sample compared to everything else. So the background is what is gray and what is colored, obviously. So, and the result for this result here showed that if you look how many sphingomyelins are here in that group, we find that sphingomyelin is obviously enriched, but also other things that we haven't actually noticed up to now. For example, fatty acids with 14 carbon atoms seem to be enriched, and the LPE class in addition. So this is something new to look into. If you do the pathway enrichment analysis, we also find that there is the sphingolipid metabolism enriched. So that would give us another hint of looking into these samples and say, is there anything specific that can be used or that is useful in that pathway that we should look at? Good. So what I told you is basically what enrichment analysis is and what it can tell you. And overall, I told you about data cleaning, univariate analysis, t-tests, correlation analysis, how you actually can use a different numeric value you bring from outside the lipidome to correlate with your lipids. And afterwards, how to use these univariate and correlation, both of them are actually univariate analysis, correlation and statistical tests, and use that in enrichment analysis to find more general information in your data. And of course, statistics is only a tool to an end. So if you have a lot of rigor in the statistic method, but you don't have curiosity, you don't have domain knowledge, it doesn't make sense on a biological, medical, pharmaceutical level, then this all doesn't help. So you actually, the most important is using the tools correctly, but then also, does this make sense with your research? Does this make sense with what you're actually trying to achieve? Yeah, thank you. And then there is one more thing that we actually have a survey here, and you can actually get the lipidomics ebook and the presentation slides from this presentation if you take the survey. And you can use the QR code for that.